Hello, everyone, and welcome to today's webinar, Revolutionizing High Content Screening from 2 hours to less than 10 minutes a plate. I am Cassie Saltman of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Araceli Biosciences. To learn more, visit www.aracelibio.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you're having any trouble seeing or hearing the presentation. I'd like to now welcome our speaker, Matt Boisvert, Application Scientist with Araceli Biosciences. Matt, you may now begin your presentation.
All right, welcome to our webinar. So I'm from Araceli Biosciences, and I'm here to talk to you today about how we're revolutionizing high content screening, going from imaging in 2 hours per plate to less than 10 minutes per plate. So I'll cover today about how we're able to do that, some assay level validation, and also why you need to go from 2 hours to 10 minutes per plate without any sacrifices. So basically, high content in 2023 is slow and non-scalable. The promise of high content was that you're able to get all of this subcellular information in an unbiased fashion in a high throughput level. And that's not really the reality at this point. There's intrinsic compromise to high content imaging. So either you go fast with good image quality, but your coverage is really low. You're only getting dozens of cells per well. Or you go fast, have good sample coverage, but the image quality is not going to be there. Or if you want sample coverage and image quality, your imaging is going to take quite a long time and really not be that high throughput that you want, the high throughput you need to accomplish these large scale screens. So that's where Araceli comes in. With Endeavor, there's no need for this compromise anymore. High content is now high throughput, and we can give you speed, coverage, and resolution for these assays. So I'll walk you through how we're able to do that, introducing the instrument, detailing the capabilities of what it can do. Then I'll prove those capabilities, giving you evidence at the assay level of how we're able to do this. And then also I'll give you a peek under the hood and how we actually accomplish this and these speeds that seem like unachievable with resolution and coverage. So first, the state of high throughput, high content right now is basically a glorified plate reader or cell counter. You really can't see much level of detail if you want to go fast with coverage. You can get cells, maybe. You're not really going to be able to pick out individual cells. Maybe you're going to be able to count total fluorescence. But if you need any level of detail, you're really not going to be able to go fast with or have that coverage. So if you need to detect something at the submicron scale, here we have an example of gamma H2AX, which marks double-strand breaks. We'll talk more about that later. But the point of this, that's a submicron level assay. So if you need to detect submicron scale, you need to detect it such that you're able to get this sample coverage. You're able to get not dozens of cells, not hundreds of cells, but thousands of cells at this level. You're going to have to do something drastic, like maybe get 4 microscopes if you want to be able to screen 10,000 compounds a day in duplicate or triplicate or 4x. Because your typical high content imager is going to get you maybe 3 to 5 plates a day with decent coverage, maybe not in full well, but like maybe a couple hundred cells. You can maybe get 5 plates a day with that. Is if you need to scale up the screen, you're not going to be able to do it with that single instrument. So you're going to be able to have to make multiple instrument purchases. And then you're going to have the space for those instruments, going to have the user base for those to run those instruments. And then you have questions of, will they all give you the same results? Can you really compare between these microscopes on the same screen? So it ends up being fairly problematic, not to mention expensive there. So not only that is you need that coverage. So a lot of microscopes, if you have that single field of view with a good resolution, if you want that submicron resolution, you get around 80 cells in that field of view. Some of the bigger ones get you 350 cells, maybe you tile a little bit, get that 350 wells in the center. I'm here to talk to you that for something like a 384 well plate, you can get over 3,000 cells in that well. So this is really going to go to eliminate variability, get more consistent assays at scale without compromise. So the reason why you want this is sometimes you're looking at some of these rare events. So in this particular case, we're looking for mitotic cells. So that's going to be marked by this tubulin stain, and there's not that many of them in a single well. If you need to get good sample coverage, you're simply not going to be able to find it on these other instruments. And this is something that you need for these rare events. This could also be something like, say, a viral infection or something of that nature, where the single cells being infected are what causes a failure or a hit in your screen. And so you need that full well coverage to be able to see these rare events. Another thing could be some sort of clonal analysis you're doing, and you're like, is there any single clone that got transfected? Again, to find these rare events, you need that sample coverage. And so it's not just about sample coverage eliminating from these rare events. You also need to do things like eliminate variability. So here on the left is a sample that came from when we were getting our washing system up and running. And it wasn't quite optimized, so I ended up washing a lot of these cells off the center of the plate. In that single field of views, this is around 1.2 millimeters by 1.2 millimeters square, we have like a dozen cells there. If that was all you're taking in the assay, you could get some result, sure, which is this red line on the right, but is it actually representative? Because there's a whole lot of cells that you're missing here. So by looking at just a few cells, maybe you're biasing your sample, and that was indeed the case. If we had just analyzed that center field of view in this experiment, we get a much different answer than by looking at that full well, because there's not that many cells, little bits induce this variability. You don't really know what's going to happen here. So basically, your effect changes by more than twofold. Your EC50 value is going to be radically different because it looks like the curve flattens at first, but it actually, if you look at that full well experience, the curve isn't flattening there. So you will actually get the wrong answer by just looking at a fraction of the plate that you see. And that is not what you need. You need to be able to eliminate that variability and look at the whole sample. So similarly, a similar thing happened in this nuclear translocation assay we're doing. If we had looked at the center, we probably would discard that. There's only 6 cells. So then that assay is a failure. You can't use it. If we had looked some other imagers, you can get a couple fields of view on the sides. This is around 2 millimeters square. And that ends up getting a piece of hair, dust, whatever in there. If you had tried to measure that, you would have gotten a very different effect than what we actually see. So in red, those are those 2 fields of view that I've highlighted. And they're measuring that dust. And that dust, surprise, surprise, does not look like your actual sample. So again, if you just measured those 2 fields of view, you've gotten a much different thing than if you looked at all the other fields of view. And again, those aren't corresponding to what the replicates look like either. So you'd just be biasing your results by not looking at all of the data here. So that's why you need that sample coverage. So hopefully I've convinced you a little bit, at least, that you need both speed, coverage, and resolution in your high content scanning. But unfortunately, there's no real solution on the market right now, except for one, which is the Araceli's Endeavor. So this is really how we got to no compromises between speed, coverage, and resolution. So now I'm going to go introduce the instrument to you a little bit, tell you about what it can do, then provide some evidence that it can actually do the things that I claim. So Endeavor it's a scanner that basically has a single 20x objective on there. There's no switching there, so everything's optimized for that objective. The idea behind this is that we're able to give you optimized results based on a single light path that we can then become quite flexible with by doing things like downsampling and that sort of thing to get you the results that you need. And I'll demonstrate how it's flexible there. And so the shtick with this is that basically you can get this whole well resolution, and by whole well we mean somewhere between like 80 and 90%. We generally cut off the at very edges of the wells. You can configure your scan to get those well edges should you need that. But that allows us to get things like this overview of the whole well. But then you can zoom in on the details, and all the details are maintained throughout the entirety of the well. So you can zoom in even further and get that sub-micron level of details. Like in this particular cell, it's a little bit weird looking, but you can see that there's these actin filopodia that are at the sub-micron level that we can accurately resolve. And so this is really the main value proposition of Endeavor, is that you can get this coverage with this resolution and at speed. Because that's really the important part of this that really brings it all together, is that if you have a 96 well plate, you can get a single field of view, which is again that 1.2 millimeter by 1.2 millimeters in the center. You can get that in 38 seconds at the submicron resolution. Our pixel size for resolution-wise is 0.27 microns per pixel. So you're really able to get that submicron resolution. If you have a 384 well plate, that single field of view it happens in 3 minutes. And again, this is in four channels. If you want to go full well, we can get in a little over 6 minutes, more like 7 minutes, you can get that full well coverage for a 384 well. It's taking only 8 minutes. Again, this is in 4 channels. And best of all, you can get your 1536 well plate, which generally takes hours to image, in 9 minutes. And so this really means that you can increase your throughput massively. So basically, this gives you the speed to really accelerate your screens. If you're running this instrument 24/7 on 1536 well plates, you could potentially screen 250,000 of compounds in a single day. And that's with these 4 channels. And that's not even the same scale that other microscopes on the market are coming up with. So this really means that you could really, if you really want to hammer out this, you could run through your library in a matter of days. Or if you have a bunch of different users with a bunch of different assays, their screens are now happening in the level of 10, 20 minutes per plate or a couple plates. And then you could get a lot of different users on this instrument in a day. So I'm sure I don't have to tell you all about why submicron resolution is important, but I will anyways, because that's what I'm here for. So this resolution really allows you to get at exactly what you want to measure. So with something like this, again, this is that gamma H2AX stain marking a particular flavor of histone. And these are truly submicron level spots, as you can see with the zoom in. In order to measure something at this level, you need that submicron level resolution. Again, it's a 0.27 microns per pixel. So you can really accurately resolve these submicron level spots. And this actually matters. If you were to measure this exact same assay doing, say, just pure nuclear intensity, so you segment your nucleus, asking how much intensity there is in the green, you don't really find an answer. Aminomycin, which is a known double-strand break inducer, gets the same results as mannitol, which is just sugar. Whereas if you're now going to a spot counting assay, counting each and every submicron level spot here, your aminomycin has almost a 30-fold effect size between the control and the high dose. You can get a lovely EC50 curve, and your Z prime here is 0.87. So you have this really robust effect that you can get only by counting the submicron level spots in these assays. In addition to that, resolution allows you to get some things for free. So this is micronuclei that we're looking at. So just using this Hosch stain, which is again just this DNA stain, we're able to visualize these micronuclei, little fragments of DNA that have broken off from the nucleus and been expelled from the nucleus. And this is indicative that there's some sort of genotoxicity happening. I ended up looking at some an autophagy assay I was doing, just incidentally, that wasn't the main outcome of the assay, just something I wanted to look at. I was able to find micronuclei for a couple of compounds indicative of genotoxicity, and that even wasn't what I intended to measure with this assay. So basically, I was able to get these measurements for free, essentially, and it's due to the submicron level resolution. So what does this all look like in the real world? It's one thing to sit here and talk about some things I did in our own lab, in the safety of our little building in Oregon. but we actually took this out in the wild, and basically, in collaboration with Sonja Sievers at the Max Planck Institute, she basically sat down with Endeavor and said, how many plates can I screen? So she is mainly working with cell paint with 384 well plates. With her current HCI system, she normally does around 3 plates a day. With Endeavor, she was able to scan 48 plates in this 8-hour workday, and she stopped for lunch. So basically, she was able to 15x her throughput here. And not only that, is this data quality, so 15,744 wells scanned, with only 0.2% of those ended up being out of focus. And I'll talk about how we get these metrics in a second. But we also were able to do this at better resolution than her current instrument. With her 3 plates a day, she was looking at 0.65 microns per pixel, whereas Endeavor is getting that 0.27 microns per pixel. So again, much better resolution there. And there's improved well coverage. With her scanning, she's able to get under 2,000 cells per well there, because she was only getting 2 millimeter by 2 millimeters, whereas we get more cells per well with our tiling our fields of view. And so again, that's really going to eliminate variability. So maybe from here, you could need less replicates because you have more reliable data. The question now is, if you can produce all these images, how do you actually know if they're worth analyzing? So for that, we developed a first-in-class quality control tool. So this is actually assessing on the level of content how good your data are. So the way that we're doing that is you provide an example image. So this is something that you think your staining will look like, something that should be representative of your staining. Generally, we use nuclei for this because they're fairly reliable. We then transform those images into Fourier space and compare them against every FOV in a given wavelength. So basically, this is a content similarity statistic. So from that, it will say things like where there's like, maybe different patterns of staining, but largely the same circular staining. Great, but there's no content in that well, it's gonna flag that. If there's, say, some weird light refraction, it'll flag that. If there's some schmutz in your well, it flags that. And it'll give it assigns it a score of 100, but basically, how different does this look? So this is some real world data. So this came from Fina Vista. They were doing some neuronal work for us, which I'll show a little later. And basically, they had cells in wells K3 to K18. You'll actually notice that when they pipette, sometimes there's some splatter. So there are some cells that splatter out. So that's why you have some higher scores around the edges of that. But you'll notice that there's one well that was flagged. And so that indeed, it seems like there was like an autofocus loss. I believe there was a scratch by that plate. And so that, like, out of hand will get discarded. But there's also some intermediate scores. And these intermediate scores are detecting things where there looks like maybe there was some schmutz in that well. Maybe there is just something happened, there was a fold or something like that. And then you could then assign, okay, I'm gonna inspect anything that was between 80 and 90, or I just want to look at the most quality data. Anything over 87, I'm going to accept that. So this way, you can have this level of quality control without having to manually look through 100,000 images. Not only that is that we also can offer bright field with submicron level resolution. And this doesn't involve a turret or any sort of switching, so it literally only adds 1 to 4 minutes of extra scan time onto your images. So this is what a bright field looks like. And the cool thing is we did correlative bright field and fluorescent staining. So we can look at, say, these actin fluorescent stain combined with this, and you can see that we end up identifying some actin filopodia in that fluorescent stain. We see the same filopodia in bright field. So this really, you can actually get at some like sort of texture level of your actin here without doing any sort of staining. Similarly with nuclei, you can pick up things like nuclear texture. In this Hush stain, we find you can identify things like little craters that where nucleoli would be. These are even more apparent when you have that bright field staining. So you could really get a lot of different metrics out of this that you wouldn't be able to otherwise without any additional cost, just a minute or two of extra scan time. So with that, you could even try to do a lot of machine learning-based training on your bright field, correlating that with different fluorescent dyes, and get really powerful algorithms that without any staining can identify a lot of different things. You can also put this in line with our quality control. So if you did something like that, you could do that single preview scan, which again on a 384 well plate takes around 3 minutes. On a 1536 well plate, bright field alone will take under 5 minutes. So you could have this be a quality control check, unstained cells, put them in Endeavor. 5 minutes later, you have your bright field. You can then assess that for quality. Say, are there cells here? So basically, things in the 90s, they're cells. They look nice. But this one score in the 60s, there's no cells in that well. Something happened. There just ends up being a lot of condensation. So essentially, what we've been able to do is assess the content of the bright field images as well as fluorescent with this quality control metric, which could be an inline in your production line to really say, oh, before I spend $20,000 to stain this plate, let's actually see if it's worth staining. All right, so I have talked about what it can do and just the basics of, okay, we have the resolution, we have the coverage, we can image a lot of plates, but we need at the assay level of actually, can you show effects that are common? Can you do the common assays? To that extent, we have a little lab here in Oregon that has been fairly productive, and we tried to validate a lot of the common high content assays. So I'll first talk to you about NFKB, which is like a very canonical nuclear translocation assay. We'll do submicron level nuclear spot counting, which is that genotox that I've been showing you. We'll then look at another spot counting assay, autophagy, but this time we'll try 2 different techniques to get at the same answer to show that we have a flexible instrument and that it's not just a fluke that we can get results. We can get results through different modalities of imaging. I'll also show you some cool things about bright field in there, touch on some neural imaging, show how our light path, despite having a single 20x objective, can be quite flexible with some cell positivity staining. And then I'll touch on some textural stuff, including cytoskeletal morphology and some mitochondrial things. So the first thing up is nuclear translocation. So this is like one of the first high content assays ever out. We have a published app note on this on the website. So if you go to our website, you can see that. But basically, the basics of this assay are that the transcription factor NF-κB, NF-κB, is in the cytosol, translated, waiting for a signal, generally some sort of stressor, to come, where they then translocate into the nucleus. So this is a very common assay to look at things like stress, or basically seeing, hey, is this pathway being induced? So what we're measuring here is we basically look at the cytosol. So define it. We can define that the cell body. We can define the nuclear area, and then subtract those two to get a cytosolic measurement. We'll then ratio that cytosol to the nucleus and basically get a single number that represents how much of this protein has gone from the cytosol into the nucleus, and is it primarily cytosolic or primarily nuclear? Is it being induced, essentially? So this is an immunohistochemistry-based assay, and you'll see that upon application of cytokine, the picture I'm showing is for IL-1 beta. We also did this for TNF alpha as well, both of them very classic cytokines, so very classic inducers of the stress response, and really indicate it. So we see there's no treatment here. Basically, you have very characteristic cytosolic staining, looks kind of like a weird shaped donut. All of it cytosolic. In the intermediate dose, you have both cytosolic and nuclear staining, and then at the high dose of cytokines, it's all of the NF-κB signal, which is from this antibody, is indeed in the nucleus. So from this, we're able to get very lovely titration curves. So you'll see that for both IL-1B and TNF-alpha, we are able to match EC50s that have been previously published. It's a fairly well-known compound, and these are very characteristic cytokines for that. And so we're really able to show that, yes, we can reliably detect this translocation. Next, moving on to genotoxicity, this is a very different sort of assay. So it's also using immunohistochemistry. This is against a phosphorylated serine in the histone H2A. So this is going to be really characteristic of double-strand breaks. When there's a break, this histone gets modified to identify the site of that break. So we put the aminomycin, which also known as mitotosin C, and that essentially is a robust inducer of these double-stranded breaks. So then what we're doing is we're looking just at the nuclei and counting the number of spots in this nuclei. When there's a high dose of sugar, you don't really see much in the way of this H2AX staining. When there's no treatment, same thing. With an intermediate treatment, basically you can get some cells are high responders, some cells aren't. But basically at these, you get a really robust effect of these submicron level spots, which we can accurately count at the high doses of this double-strand break inducer. And so basically we can get that like 35-fold induction we see. And again, as I showed you before, if you're just looking at this as like a nuclear positivity assay or like some sort of intensity-based assay where you're not actually counting these submicron level spots, you really don't get an effect size here. Whereas here, we're able to get a Z prime of 0.87, which indicates a really robust, not particularly variable effect. And we're able to do that with scanning 85% of the well area of a 96 well plate in 6 minutes and 40 seconds for these 3 separate channels. So really able to get submicron resolution really fast. Next up is autophagy. So autophagy is basically the cell's way of dealing with internal garbage. Autophagy essentially means self-eating. So what we're looking at here is you give some insult that will induce autophagy or block autophagy, and then measure how much of these autophagic vesicles are accumulating. So with that, there's 2 different ways to go about doing this. They're both like robust commercially available kits. One is the immunohistochemistry. I've shown you a lot of immunohistochemistry already. So this is against the marker LC3B. It marks autophagosomes preferentially. It's readily accumulating in the nucleus in the absence of autophagy. So that means we just have to look at cytosolic accumulation. And again, we have to do the spot counting rather than a total intensity thing, because in just a basal state, you'll see that we have some amount of intensity for that green channel without any treatment. So we're really just looking for accumulation of these spots in the cytosol. However, immunohistochemistry ends up being kind of expensive. It's a little tetchy sometimes. It's a longer staining process, and by and large, you need fixed cells for this. So with that, we also looked at a different modality, which is a live staining. So this is, I believe, from Enzo. It's a cationic amphiphilic dye, which basically means that they figured out the conditions of these vesicles with very particular pH and lipids, and so the dye only binds to that. So it's selectively binding to these autophagosomes. And the nice thing about this dye is it's staining live cells, but the not so nice thing is it can't be permeabilized. So essentially, these are 2 very separate techniques. We'll see if they can get us the same answer. So first up is that antibody staining. So basically, we're quantifying those cytosol exotics. The way we're going to do this is we'll segment the nuclei, because again, we don't want that nuclear signal in there. And then we'll segment the cells themselves. This is based on that phalloidin stain. And then so with our cellular segmentation and with our nuclear segmentation, we can make that cytosolic mask, just as we did for NFKB. Then with that mask, we're just asking where there are these spots in there. And again, these spots are around a micron, maybe a micron and a half in diameter. So you really need that resolution to accurately count them. And so with that, we looked at MG132. So this is a proteasome inhibitor So essentially, it arrests the autophagosomes, so you see them really accumulating in that cytosol. And that is indeed what we found. So with the LC3B alone, you get that characteristic intense nuclei that we don't want to measure. At an intermediate dose of MG132, you find that there's some dots, and then essentially with that high dose, there's a lot of spots. Great, we can get a really robust Z prime score of 0.83 out of that. Similarly, we induce the autophagy through mTOR inhibition. This is Torin 1. So this is now using that cyto-ID dye. So same thing. With an intermediate dose, there's a lot less nuclear staining here because, again, LC3B is actually in the nucleus, whereas this is now doing a lipid-dependent dye. So a lot less of that nuclear staining. So this is just raw counts of the cyto-ID dye. And basically, in an intermediate dose, we find that there's some dots. We get really bright, chunky staining at the high dose of Torin 1. So with this, you can get a really lovely curve. The EC50 value here is 0.1 micromolar. The Z prime of 0.82, again, indicating a really robust assay. And again, for that 3-channel 96-well image, which ends up being 4,608 images, we're able to get that in 6 minutes and 9 seconds. So again, this is a really fast, robust screen here. So the next question that I really want to ask from this is, can the dye and IHC give us the same results? So this is using autophagosome degradation inhibitor, hydroxychloroquine. And so up top is our antibody staining. So you'll see the antibody staining, again, characteristically nuclear with really well-defined spots. And you see a really robust effect. I mean, the effect size is massive. With the live cell dye, it's a little less vibrant, again, because it's not immunohistochemistry. Immunohistochemistry is really the gold standard for a lot of these. So again, this is measuring those aggregates per cell. But again, you still get a really robust response. And with good Z primes for both assays, the Z prime for the IHC is better, of course. But then you also have a really well-matched EC50 values. So you have an EC50 value of 5 micromolar for the IHC and an EC50 value of 3.5 micromolar for your live cell dye. So again, this is really nice because it shows that across these modalities, we can get the same results, and they're consistent and they are robust. You can even see these autophagic flux differences in bright field. So this is using the LC3B dye. This was an older plate, so the staining is not quite as nice, but I ripped off that cover and put it under the transmitted light as well. So we can do this, so you can really see in here that there's, you can see the autophagic vesicles there in the bright field. So you can do the overlay, so you have the overlaying of the LC3B, and it's co-occurring where you can see the obvious vesicles. So again, this really gives you the option of doing something like training a machine learning module here. And that machine learning module will be able to automatically recognize those vesicles based on the staining. So then you can do your first set of screens with both bright field and the LC3B IHC, then train your model on that, and then you can go in and basically go do the rest of your entire screen as just that bright field, because now you have a model that's trained to recognize those vesicles, can robustly recognize those vesicles, and you all of a sudden just saved your lab $100,000 in your massive screen, which is great. Another thing that I always like to look at is neurons. I'm a trained neuroscientist, so I'm always really happy to see them. But looking at these full well stains is really the good way to look at these neurons, because you see that they have all these connections, and the connections don't stop where your field of view ends. So you really need that whole well imaging if you want a good sense of what these neurons are doing. So we can zoom in a little bit, and we just treat with cytosporine, which I believe is an ATPase inhibitor. You basically are wrecking the connections. You don't see that many connections. You're inhibiting growth. With low doses of cytosporine, you have really robust growth. Great. I haven't gotten around to quantifying these yet, but that's on my to-do list, so keep an eye out for that in the future. But the one thing I really like showing off on this is that this is a great usage of maximum intensity projection. So basically, you can restore all a lot of the Z-dimensional data here. And so that Z-dimensional data ends up being really important because generally you're growing these neurons in some sort of matrigel or something of that sort, where the neurons are putting their processes in the Z dimension. And if you're just imaging a single plane, you're going to miss all that. You're going to get a lot blurrier staining. So it's important to have the ability to do those max intensity projections. But unfortunately, if you're doing a whole well image with multiple Z images, so here we're doing 4 slices, it's going to be really slow. But that's not the case with Endeavor, because Endeavor is able to image at such a fast speed, and that's actually fast. It's actually quite fast with Z-Stack, so there's less movement there. So we can get the image is 384 plate. That whole well, these are low volume 384 plates, so this is actually a true whole well experiment. So we're getting 4 1.5 micron slices, 284 plate in 15 minutes and 32 seconds. And we're able to automatically make that maximum intensity projection. So basically, it's storing the Z-stack in memory and then collapsing that and projecting that Z-stack for you there. So that essentially means that we're able to get you that Z-dimensional data at full well, at the same time that a lot of other instruments on the market will get you a single small field of view, which is quite impressive and exactly what you need for applications like neuroscience. This could really revolutionize your neural screening, because all of a sudden, what used to take you 4 hours to do whole well Z-stacks now takes you 15 minutes. Changing subsets a little bit, this is also a fairly flexible instrument. I keep alluding to the fact there's that single optical path of 20th objective, and that's really overkill for something like live dead. So we did this live dead assay, which is a very simple nuclear positivity assay. It's so simple that I'm not even showing you zoom ins. I'm just showing the full image. And so basically, when this cell is dead, it'll be green. So we treat it with phenylmycin, which is very well-known apoptosis inducer, and basically we found a very lovely curve, just either treating the cells as induction assays, so basically above a threshold, we're calling that a dead cell, or just segmenting nuclei and then saying how much green is there. Either way we do it, the EC50 value we get from this curve ends up almost being identical to that from the kit manufacturer. Because the kit manufacturer is like, oh yeah, we tested this on phenylmycin, our EC50 is 25.5. Well, our EC50s are 26.7 and 26.3, depending on how you're doing this analysis. And that Z prime is 0.95. Again, it's impressive, but not that impressive because it's a live dead assay. Real basic. So it's so basic that you don't need this resolution. So this red line here is a 4x4 bins. So this ends up being, so that single 0.27 micron pixel, we're now summing those across 4 pixels by 4 pixels at the level of that CMOS camera. So again, this is able to get this whole plate scanned in 3 minutes and 30 seconds. That's about a minute better than we're able to do for that prior stain when you had no binning. So the EC50 is again very accurate, and again you're able, because you're doing this pixel sum, you end up getting about a tenth of the exposure time or better. So you have much more intense, robust staining here. And you have the ability to do this binning, so that way if you have data constraints, if you just simply don't need that resolution, you don't have to use it. So you have that flexibility to do what you need to do. You can also have the resolution to pick up things like textural measurements in the cytoskeleton. So again, this is an assay that just came off, so I haven't been able to analyze it properly. But we're basically looking at microtubule standing on top or actin standing on the bottom with known elongation blockers, stabilizers, or just controls. And so you can see by eye that there's a lot of different textural differences when given the different cytoskeletal drugs. So again, haven't quantified this yet, but it's looking really promising that we can pick up these submicron level textural differences, which is similarly done with our mitochondrial disruption assay. So that again, is treating with FCCP, which I believe is an electron transport chain disruptor. I believe it disinhibits the EDC, maybe. Don't quote me on that mechanism. But basically, what we see is you're disrupting the mitochondrial membrane. So the control is nice and happy. You get these long mitochondrial networks. The intermediate dose, you start seeing the networks being a little less elongated. But then at the high dose, you're really seeing the mitochondrial just ball up. So again, you need that submicron level resolution to be able to detect that your mitochondria going from these extended networks to these little balls. So hopefully I've shown you a good amount of validation and flexibility in the instrument at the level of the assay. So we can do things like that subcellular translocation, up to like different kinds of spot counting, going to submicron level spots, just slightly larger spots in the cytosol. We can look at neurons, which is a great use case for that, both the whole well and the ability to make those max intensity projections. We can also just save Z-slices if that's what you're interested in. But we can also do the low-res, really bread and butter assays like that live dead or other cell positivity assays, and also look at texture. So with that, we also have a flexible output. So this, we're designed to be able to take your data anywhere. So you can stay within our ecosystem. We're going to debut an analysis tool quite soon. All of the data that you've just seen analyzed was in an internal version of that analysis tool. But also, we just output everything in 16-bit TIFFs. We also have all our metadata just being populated in a JSON file. Again, it's designed to be open and eminently parsable by pretty much anything. So this really allows you to basically be like, okay, I want to do X, Y, or Z with my data in this other program. You can. And you're not locked into our own internal systems. So now we get to the fun part, which is where I present you all this data and I'm like, oh, by the way, you can image this in 6 minutes. You can image this in 7 minutes with the whole well at submicron levels. And so at this point, you're like, well, how is that possible? So I'm going to actually show you the instrument action and talk about what's actually going on here. So basically, how are we able to collect 5 channels of data, so 4 fluorescent plus bright field, for 1536 wells with submicron resolution, getting 82% of that well area in under 10 minutes? Well, now we'll go live and see how I'm able to do that. So this is the instrument here. So we have a plate. So this is our standard plate. The instrument is indeed a tabletop-based instrument, which is great. We also have an optional robot with it that can automatically load and unload plates, as well as read barcodes and then load those off of the barcode. But I'll spare you most of the demo here to just load this plate, and then we can sort of see what's going on as I image it. So now I'm going to switch to doing a screen share. And this is going to be the instrument that is being run remotely. So that's that instrument I looked. So I'm going to close the door now. And so you'll see that you can load protocols. This could be based on those barcodes or something like that. Or you can start a new protocol here. You have our 4 channels. And so basically, so hopefully this is set up well already. So I'll just be able to put on that preview mode and start scanning. Yes, so you'll see we have these lovely images popping up. And so the thing that I really want to highlight here is that we're populating these 4 at a time. So basically, that's one of the main ways that we're able to get the speed that we're able to get. Pardon? The speed that we're able to get is because we have 4 independent optical paths. So each one is a 20x objective with its own optical path. And so by doing that, we're really able to image basically 4 times as fast. So I'll talk a little bit later about how else we're able to get our speed, but that's really the main point here, is that we're really take that single optical path and 4x it and put it in that box. And so you'll see here, that I just scanned out live, you can get a really lovely nuclear texture. You can see those individual actin bundles as well as some filopodia. And so this is, I believe, a WGA stain, so we're able to see a little bit some of those membranes. And then also on there we have far right, so this is a mitochondrial stain. And so we're able to get this live. And so I'll turn off preview and start another scan so you can see what that looks like as a full well as well. So you'll see that it's operating as a mosaic now and imaging around. And so again, this is what we call our full well scan. It chops off the very edges here, but it ends up getting something about 85% of that well area. Okay, and again, let me know afterwards if you'd like to see a fuller demo, and I'm happy to go that. All right, so now back to the presentation. So you saw that we have these 4 independent optical paths. And so those 4 independent optical paths are very important to getting our speed, but that's not it. That's not all of it, because that would only get us 4x speed, and we are operating at the level of 10x or beyond for what we're able to do. And this is also because there really hasn't been a new 2.5D high content imager for well over a decade. So we're able to do this ground up design with a single fixed optical path, leveraging the best of new technology with no moving parts. So that's really important, is this no moving parts. So basically the only 2 things that move here are the stage. The stage is moving above the 4 optical paths. And so that's one thing. And we also have the focus, obviously. So you're able to focus those 4 the 4 objectives. But other than that, there's no switching, there's no filter set switching, none of that. So you don't have to wait while things switch. We also are using the state-of-art LED excitation. So this was designed with LEDs in mind the entire way through. This company actually came out of an LED company. The company had really good LEDs and wanted to get a good biotech basis for it. And so they spun off Araceli on the basis of this really fast, precise LED tech. We also have an optimized hardware-based autofocus. It's a laser-based autofocus that has a very clever algorithm behind it. So just basically always on and always focusing. So really able to save a lot of time there. We also are using this extra large CMOS detector. I believe it has a 2.7 micron pixel size. And it's 4,400, our fields of view are 4,400 pixels by 4,400 pixels. So it's very large, so you're able to encapsulate a lot of the well area in that single image. And again, that's adequate to image, I think it's 85% of the well area of a 1536 well with that single shot preview, which I was showing you for that 96 well plate. So in summary, I hope to have shown that I've told you how you can eliminate variance and find rare events in your samples. That's imaging that full well. Again, you can't usually do that without sacrificing image quality and speed. We also showed you a solution to how you can resolve these hard to find objects. So that could be something like a sub-micron level spot to something like a neuronal projection that you need whole well and you need Z-stacks of that. And you also just need the resolution to resolve those dendrites and the dendritic arborizations there. So you really need that ability to get those hard to find objects in there. And then, most of all, you have to be able to scale all these rare events finding, these sub-micron level spot detection up into that large scale screen. So I really hope that I've shown you that with less than 10 minutes per plate, with 96, 1536, and 384 well plates, with 0.27 micron per pixel resolution and that full well coverage, we've really solved this triangle of compromise. So you really no longer have to compromise your scanning to something, or to like downsampling, or to only getting a couple dozen cells if you want to scale up that large scale screen. So with that, I want to thank you all for listening and attending. I want to thank our outside collaborators So this is Sonia at Box Planck, Erica and the Fina Vista team. All of the lab work you saw pretty much was done by one man, Josh, working in a small lab. He's done some great stuff. And of course, I've had a lot of support within the company. And with that, I'm quite happy to answer any questions you might have. Great, thank you.
Thank you, Matt, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many of your questions as we have time for today. Let's go ahead and get started. Our first question asks, how do you deal with the sheer quantity of data Endeavor is able to generate?
Yeah, sure. I mean, that's going to be a problem because it's basically you're just moving the bottleneck down the road a little bit. So we have a couple solutions to this. One is you can just, if you don't need the resolution, you can just bin that data. So that's going, so if you do a 2x bin, that will reduce your data by 4 times, or you do a 4x bin, that's a 16-fold data. So that's fine if you don't need that resolution, but if you do need resolution, what we have a solution there. We have both fiber optic cables and copper coming out of the back of Endeavor. And we have a solution where we have a NAS, which is basically a mini server that you can connect that to, or you can upload onto your own server, so you can write directly to that multi-terabyte hard drive. And again, we offer that as a full solution, so you can then immediately write to that larger storage space there.
Great, thank you. Our next question here asks, is your microscope compatible with robotics?
Yeah, again, you actually can see right behind me, there is the robotic arm. So we worked with to get the CELA certification there to basically have that open format to support laboratory automation. We've also worked with Green Button Go with a couple of our clients to integrate into their existing robotic line, and they've been able to have good success there. So basically, we have a hotel that we can the arm can load and unload. There's a scanner there that also can read the barcodes and load protocols based on those barcodes as well.
Excellent, thank you. Our next question here asks, will the QC algorithm detect differences in my treatment groups and flag false positives?
So it is a score out of 100. So if your cells do still look like cells, they'll score fairly highly on that. So basically something that looks like a cell, and you feed it with an image that looks like a cell, they'll look pretty similar than say like an autofocus or an absent or some sort of like hair. So still, because it's not a binary yes, no, you can set that threshold there. So you can pretty much say, okay, as long as they look vaguely like cells, they'll probably score above like an 80 versus an out-of-focus well, which scores around in the 50s. Piece of hair scores around like 60s or 70s. So you're really able to move that threshold to where you want to accept or reject your images. So if you have a really wacky looking phenotype, you can just put that threshold down and basically be like, okay, we're willing to accept maybe a lot more variation in our images. But again, if you're really stringent, you know that you're not expecting like massive deformations in your cells. And again, we were generally doing this QC on the nuclear channel, so we're not really expecting much in the way of radical variation there. You would find that it's as long as like you're not, as long as the cells aren't totally strange looking, they will score normally within the rest of the cells. But again, if you're looking at a different staining beyond nuclei that is expected to change a lot, then maybe yes, the QC scoring will score those cells much lower, and then you'll either have to, then the utility is a little lessened on other stains besides the nuclear stain, or you just have to set that threshold lower.
Excellent, thank you. Our next question here that we have asks, can I image slides with tissue samples in Endeavor?
Not at this time. We have a prototype slide holder that we can use in-house. but the instrument's really designed to just work with high content plates. It's maybe in the future we're exploring something like that. In principle, there's nothing that stops us at this point, but they would take some work on the software side to make that happen. And there's no current plans to support that. If there was interest, we can certainly engage in that conversation. But right now, the answer is no.
All right, great. We have a couple more questions here before we end the webinar. So our next question asks, you showed a fair amount of analysis there. What did you use to analyze your plates?
Yeah, so it was almost entirely done on our in-house analysis software, where it's currently in, we're about to ship it into beta into into select customer hands. So it's all using in-house algorithms based on the standards in the field. So that's a lot of things that are coming from Cell Profiler, integrated with some more modern like AI-based and machine vision-based tools. And so hopefully that will be debuted in February to the general public, so they'll have access to all those tools. Because what we've essentially discovered is that by speeding up your imaging radically, you've sort of just moved the bottleneck down the line. So if you don't have a really fast way of analyzing these things, all of a sudden you have all these images just piling up. You don't have that analysis for them. So we are going to offer this. We have this analysis software package that we're going to be offering circa SLAS in February.
Great. Our next question here that we have asks, what objectives does Endeavor have?
Yeah, so we only offer a single 20x objective. It's from Nikon. It's what's called a high dry objective. So this is an objective that has a 0.75 numerical aperture, so very high resolution. But because no one really wants to deal with a water objective, it's entirely an air objective there. We only offer that single objective. We don't swap it out, but you can again do things like binning to downsample that resolution. And because we're combining this with this massive CMOS detector that has, I believe, a 2.7 micron pixel size, we're really able to get resolution that's equivalent to like something like a 30x or 40x objective with this. But then you can downsample that into something that looks more like a 10x or 4x objective. And so with that single objective, we're really able to offer a lot of flexibility. And again, we have this optimized light path just for that single objective, which again is operating parallel. So really to offer the most efficient light path with options for flexibility on either end.
Wonderful, thank you. And it looks like we have time for one more question here today. That question asks, can you image T cells and other non-adherent cells?
Yeah, so that one's a little trickier. So something, if basically this was designed for what we call 2.5D. So it's like 2D that lies flat, but we can image somewhat above that single plane of focus, probably around about up to 200 microns if there's not too much optical scattering. So the short answer is yes, as long as your cells readily are able to lie flat enough down. We don't have anything that can seek out the cells in the Z dimension. Basically, you can image through a Z plane and detect non-adherent cells that way. And if your cells are lying more or less flat on the bottom of the plate, that'll work really well. We've imaged things like T cells and PMBCs before to great effect because they're able to settle down and lie flat. The movements on our stage don't disturb them. So we're really able to image things like those T cells. But if you have cells that are really in suspension and maybe 200 microns just all throughout that column, that'll be a lot more challenging because we don't really know where they are in the column. So you can, in theory, image through that Z stack, but it's going to be a lot of data, and it's going to be a lot of data that you don't particularly need. So we can image things like T cells, things that lie flat. We can sort of image things in a column, but that is not really designed to do that. So it won't necessarily be the optimal solution for that.
Thank you again, Matt, for your time today and your important research. We would also like to thank LabRoots and our sponsor, Araceli Biosciences, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions that we did not have time for today and any submitted during our on-demand period will be addressed by our speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everyone. Goodbye.